At the outset, I wish to thank the organizers for this invitation to participate in this webinar and review our research. In this presentation, I will summarize some of our published research and discuss the evidence that supports the hypothesis that naturally occurring agents may function as testable alternatives for therapy-resistant breast cancer. Advanced breast cancer with distant metastasis represents one of the major causes of mortality in women. The American Cancer Society has estimated new cancer cases and cancer-related deaths for 2021. The conventional treatment options for breast cancer include chemoendocrine therapy and molecularly targeted therapy. These treatment options have some limitations that include systemic toxicity, acquired tumor resistance, and emergence of drug-resistant stem cells. Because of these limitations, certain alternatives need to be thought of. These alternatives may be natural products which are less toxic to conventional drugs. Natural products such as dietary phytochemicals and nutritional herbs have documented preventive efficacy in preclinical models for breast cancer. Tabebuia avellani, or TA for short, has modest evidence for preclinical and clinical efficacy against organ site cancers other than the breast. Tabebuia avellani is a tree whose inner bark contains the active substance. This tree is indigenous to South America, Amazon rainforest, and is routinely used as folk medicines. Tabebuia avellani has two active agents. One is naphthofurandione and the other one is beta-lactine. The study objectives are the following. We are interested to examine the growth emission of our models by Tabebuia avellani, identify possible mechanisms, and identify susceptible molecular targets. In this study, we have used 4 cellular models. The 184B5 is used as a control. This is a non-tumorigenic cell line. The MCF7 is a tumorigenic cell line that expresses estrogen receptor alpha and progesterone but lacks the expression of HER2. The MCF7 arome cell line is derived from the parental MCF7 cells that have been transfected with aromatase gene. The MDA-MB231 cell line lacks the expression of estrogen receptor alpha, progesterone, and HER2, and therefore is called as triple negative. We have looked at the growth pattern of these cell lines and have shown that the process of cellular apoptosis is increased by 4 to 6-fold in response to treatment with TA in these models. In the MCF7 model, we have looked at gene expression using microarray analysis. The human genome slide of 4 by 44K format was used. We have looked at differential gene expression of almost 44,000 genes. In this slide, I have selected some of the genes which are directly relevant to our research. We have looked at the modulation of gene expression. The upregulation or downregulation greater than 1.5-fold was significantly different at the p-value of 0.01. In general, the genes involved in apoptosis were upregulated, and those involved in estradiol metabolism. The genes involved in proliferation, gene for ER alpha, were downregulated in response to TA. In the MCF7 ARO model, we have seen the induction of caspase-3,7 activity in response to treatment with TA, and in parallel, the expression of pro-apoptotic BAX was increased, whereas that of anti-apoptotic BCL2 was decreased. In addition, the several estradiol-regulated genes such as ESR1, gene for ER alpha, aromatase, and PR were downregulated, while PS2, GREB2, and cyclin D1 were also downregulated. In the MDA231 model, abnormal RB signaling is documented, and this results in loss of normal tumor suppression of this gene, by this gene. We have seen that in response to treatment with TA, cyclin D1 and PRB is to RB ratio was downregulated. This ratio was predominantly downregulated because of inhibition of phosphorylated RB. As far as the apoptosis is concerned, we have seen upregulation of sub-G0 population and upregulation of pro-apoptotic caspase. The present evidence supports the following conclusions. TA inhibits hyperproliferation via cell cycle regulatory and E2 dependent gene expression. TA induces apoptosis via apoptosis specific gene expression and thereby validates a mechanism for identification of efficacious cancer selective natural products as alternatives for therapy resistant breast cancer. We are grateful for research funding from Tabebuia Japan company and from the philanthropic support. I will close here and take questions.